Sure. Many of you may know Dr. Alexander, but for those of you who do not, Caleb is a professor of epidemiology and medicine here at Hopkins. He also serves as the founding co-director of the Center for Drug Safety and Effectiveness and the PI for the Johns Hopkins FDA Center of Excellence in Regulatory Science and Innovation. He's a practicing general internist and has completed seminal work in pharmacoepidemiology and drug utilization. I would be remiss to not mention that Dr. Alexander received his BA cum laude from University of Pennsylvania and an MD from Case Western, as well as a Master's of Science from the University of Chicago. Dr. Alexander has published regularly in leading scientific journals for general mass media, garnering much attention for issues related to opioid use disorder. He's also a frequent speaker on healthcare issues, most notably and relevant for today's talk. Dr. Alexander he served on numerous editorial and advisory boards, including the US FDA's Peripheral and Central Nervous System Drugs Advisory Committee. And with that, welcome, Caleb.
Great, thank you. It's great to join all of you. And because I'm a pharmacoepidemiologist, I always hold the Center for Clinical Trials and Evidence Synthesis close to my heart, of course, where clinical trials and pivotal trials often leave off, observational work picks up. And so there is a lot of synergy between the efforts of many of you and the sort of work that I primarily do, which is focused on observational evidence. Just by way of disclosures, I'll just give you a chance to see this. I think as probably most relevant is my role within the FDA's Peripheral and Central Nervous System Advisory Committee, which reviewed Aducanumab. And I just want to also give a one quick infomercial for the Center for Drug Safety and Effectiveness. Again, I think many of you probably are familiar with our center. We welcome you to join our listserv. We won't spam you out. And take a look at our website, which includes information about programs for trainees, some information about different data assets that we have available, and importantly, a roster of faculty that are interested in drug safety, drug effectiveness, and broader topics in pharmacoepidemiology and pharmaceutical policy. So we look forward to engaging with you in any way that we can. So a few of you, I hope not all of you, but a few of you have probably heard me talk about the case of Aducanumab, although frankly, it's a case that continues to unfold, and my title of One Surprise After Another continues to be the case, including a resounding rejection of the product by payers and by health systems alike. But I think it really is an opportunity to sharpen your saw and to consider some of the many topics that are important in the work that I know that many of you do day in and day out. Whether we're talking about evidentiary thresholds for access to market, and what constitutes quote unquote substantial evidence, which is a regulatory and legal term of art, or whether we're talking about accelerated approval. We'll get to that shortly, so let's put a pushpin in that one. Or whether we're talking about futility analyses. And frankly, I would love to hear from some of you your thoughts about futility analyses as they apply in this instance. And I am going to make a commitment to myself to end by 10 minutes after the hour. I want to leave us at least 20 minutes to engage, but I also am a big fan of the chat function. And so chat me up, chat each other up. Anything I say that gives you pause or outrages you or surprises you or anything in between, please feel free to put your thoughts in the chat function, and let's use that as another way that we can communicate. I don't have the benefit of being able to monitor it continuously, but all of your co-participants do. And you're welcome to engage with each other. I won't be upset. And it also sort of preserves a record of things that we should come back to as we proceed. By the way, random high bias, is that something is that a term all of you are familiar with? The drug label, we'll get to that shortly, and that's also important. So this case of aducanumab, this curious case and I'll give you a brief regulatory history. So this is a product that was developed based on the amyloid cascade hypothesis, which posits that the deposition of amyloid in the brain is causally implicated in the pathogenesis of Alzheimer's disease. There were two nearly identical trials that were performed testing this product. There was a determination of futility that was made. Further analyses were performed. One trial was negative. The second trial showed a modest treatment effect in one of two treatment arms. There were many post hoc analyses done. And there was an advisory committee that I was a part of. There are safety concerns about the product. We'll come to those as well. If I refer to them as ARIA, that stands for amyloid-related imaging abnormalities, ARIA. So the primary safety concerns include both microhemorrhages in the brain as well as brain edema or swelling. And that's because this same amyloid that this product targets is present in the walls of blood vessels in the cerebral vasculature. So we have ARIA as the principal safety concern. And of course, the product was approved in June to much outcry and consternation and the like. So let's look for a minute at some of the results of the trials. And let me say first about futility, by the way. So there was a decision to declare futility, and then that decision was essentially backpedaled. And so I don't want to go down rabbit holes because I am trying to cover a lot of waterfront in 30 minutes or less. But the bottom line is that that per the FDA and the sponsor, the reason that the decision was made to reverse the determination of futility was twofold. First, that one of the reported assumptions to determine futility was that there was not significant heterogeneity between these two nearly identical studies. And so the futility analysis was based a priori on using pooled data. And in fact, as we'll see, the two studies, the two nearly identical trials yielded very different results. And the second assumption was that treatment effects wouldn't change substantially over time. I don't know why this was an assumption, because there was a smaller dose ranging study that was designed primarily as a safety and tolerability study that found that there was a clear dose and time dependent effect. And I quote there, a clear dose and time dependent effect of the product. But in any case, and we can come back, I'm giving you 20 minutes for us to converse. So, and if the chat hasn't already lit up, if any of you have questions or thoughts, I see only one little red comment in the chat so far, and it's probably an administrative one about, getting credit for attending the talk or something. So if any of you have thoughts about futility or know more than I do about whether or not this was a reasonable determination or not, by all means, I'm happy for you to share those with us. All right, so let's look then at some of these data. So the first trial, so study 302 was the study that showed an effect in the high dose arm. And what you see here is about a 22% reduction in 18-point scale, the CDR, which is a clinical dementia rating sum of boxes scale. And essentially the high dose group did 0.39 points on this 18-point scale less worse over time. In other words, they worsened at a slower rate than the placebo group. And if you look at these various components, or I'm actually not clear if these are components of the scale or other dimensions that were assessed, but you see effects on memory, orientation, judgment, problem solving, community affairs, home and hobbies. They're all about the same magnitude, and they're all in the same direction. And I don't know, you tell me, is this additional supportive evidence, as some have suggested, or are these correlated and essentially capturing the same changes that we see captured by this overall summary effect of 18 points? So maybe the product looks okay if you think about this trial. And these are the results for this same study over time. And thank you, whoever threw me excuse me, I'm assuming one of you threw a comment in the chat. So I'm feeling my spirits are a little more buoyed in seeing something there. But in any case, these are the results over time. And what I would point out is, look that there's no separation of these treatment arms until the 18-month endpoint. That is, there's no separation early or sort of during the midpoint of the trial. It's only at the 18-month point where you start seeing separation. And note also the number of missing. Because futility was determined, essentially, what happened was the manufacturer went back and continued collecting information about everybody that had been enrolled at the time the futility determination was made. But you nevertheless end up with a lot of missing data. Well, you tell me, you guys are the trialists. But 45% missing data to me feels like a big number. So if it's not as big a concern as it might be, I'm all ears. And what about the nonlinear disease progression? So this is another one that's maybe a little bit of a rabbit hole. But I spoke with a smart group of I don't know if they were molecular biologists or something the other day about this, and they said, well, is the CDR-SB a linear scale? And the reason this is important is because of when we start talking about what the minimal clinically important difference is on the scale, and we start comparing that to the change that was observed in the high dose treatment arm at 18 months. So just to cut to the chase, the minimally important clinical difference on this 18-point scale is, what do you guys think? Maybe throw it in the chat what your guess is as to this. So there's an 18-point scale that measures how severe your Alzheimer's is, or your mom's Alzheimer's disease is. And it's an 18-point scale. So what's the minimum amount that you think, if you go to patients and providers and say, can you tell us what you think the minimum amount is that you would need to see on this scale in order to be convinced that this is clinically important? Is it 1/4 of a point? Is it 7 points? What do you guys think? So this sort of complicates matters, and thanks for those of you that are chiming in there. We'll come back to that in a minute. And now we're getting into a little bit more of the interesting nitty-gritty here for the trialists in the room. Could placebo separation explain the results of the positive studies? So there was an amendment to study 302, and the difference in the placebo and the difference in the placebo response before and after that amendment was very noteworthy. And in fact, the statistical reviewer at the Food and Drug Administration has helped us and noted it here. And he raised concern that in study 302, the positive study, in fact, what you see is that the effect of the dose increase that took place because of an amendment at some point during the trial, and I am going to come back to what the minimal clinically important difference is, but the effect of the dose increase in this after this amendment is entangled with simultaneous placebo worsening, and which raises the specter that in fact the positive result that we saw just a few slides ago is not due to the that this conclusion that the high dose treatment arm has an efficacy signal may actually not be the treatment working better, but it could be the placebo working worse in a subpopulation within study 302. Oh, and then there's the negative study. So, I suppose, other than that, Mrs. Lincoln, how did you enjoy the play? But you have another nearly identical study that not only didn't show a strong signal, in fact, the high dose arm did nominally worse than placebo in this study 301. So you have a whole number of complicated issues that I suppose put you and me in business. And during this advisory committee meeting, there was a very smart biostatistician, Scott Emerson from University of Washington, that I believe his first comment was using the Texas sharpshooter fallacy, and just pointing out that that fallacy, of course, is about a guy that goes and shoots up a barn and then paints a bullseye around the holes that he likes the best. And I think the issue here is can we, after the fact, confidently explain sufficient for the FDA statutory requirements of substantial evidence of safety and efficacy, can we explain how on earth we ended up with two trials that point different directions? Could it be due to baseline differences? Could it be due to protocol amendments? There was some that suggested a theory of rapid progressors, which sounded to me strikingly reminiscent of another committee, in this case, one that I chaired on eteplirsen, the drug for Duchenne's muscular dystrophy, where some at the committee hearing talked about super progressors or super responders. I'm sorry, these were not super progressors, these were super responders. And the person in that setting was interested in whether or not the product might be especially effective in the subpopulation of super responders, and how should we take that into account so as to not, throw the baby out with the bathwater. We talked about lots of these, and if there's interest in the 20 minutes that we'll have together, I'm happy to address any of them. But the bottom line is that no advisory committee member was convinced that there was substantial evidence. And I think many of us were concerned about post hoc explanations for uncomfortable realities that, in fact, we just don't know why we got discordant results from two similarly and well-designed trials. Does the magnitude of the statistically significant effect matter? So the minimal clinically important difference is 1 to 2 on a scale from 1 to 18. So you have a product that achieves 0.39 after and only after 18 months, and you have a minimal clinically important difference of 1 to 2. Interestingly, and sort of in my mind, inexplicably, although the FDA may well have a more, I'm sure that they have a more recent explanation than what's been provided, they allowed for any statistically significant difference in the outcome to suffice as proof of efficacy. In other words, they didn't say you have to raise the bar any higher than simply showing a statistically significant difference in the outcome, in the primary outcome, the CDR-SB. So not sure what to do about that. And then, of course, there's the safety concerns. All right. So this is a seminal article. I imagine many of you are familiar with it, but if you're not, go take a look. There's a reason it's been cited. I don't know. I presume it's been cited 1,000 times, maybe not. But in any case, it's a good read. And as Tom mentions here, a major source of bias in healthcare research comes from the interest in finding evidence suggesting that an intervention for treatment or prevention of disease has a favorable benefit to risk profile. I take it none of you are falling out of your seats. I only see some of you on video, but you all seem to be staying put. So this is not controversial to a trialist, I presume, especially not one at a place as good as Johns Hopkins. So, all right. So the FDA approved the product, and kind of here we are. So they approved it using accelerated approval, and they argued essentially that amyloid was a reasonably likely surrogate. Now, I didn't tell you at the outset, but in fact, there have been more than 24 products that have been failed or abandoned that have tested the amyloid cascade hypothesis, some of which have convincingly lowered amyloid levels. So it's not like we don't have a history here, and Aducanumab's own history raises significant doubts. So study 301, remember the failed study where the high dose treatment arm did nominally worse than placebo? So in fact, in that failed study, there was a strong dose response with amyloid. And yet you have the FDA's, and yet no benefit, of course, in terms of cognition. You have the FDA statistical reviewer saying it's not clear there's any linkage between reduction in plaque and long-term clinical change. You have no evidence of efficacy after 18 months. Usually when we use accelerated approval pathways, it's when we have a surrogate measure, but we don't have the benefit of having had the studies to demonstrate that the surrogate measure corresponds with a meaningful clinical outcome. And in this case, in fact, You have the clinical studies, they're right in front of you. I just showed them to you. So if in 18 months we can't convincingly demonstrate that this product works and fulfills and has enough evidence to satisfy the standard regulatory threshold for approval, which is substantial evidence, what are we going to do? Study for 24 months? And is the thought that if we just continued for 6 more months, we'd have the data that we don't have now? So, another classic by Tom Fleming, in this case, David Demetz as well. And this also should be old hat for you guys. And, for the trainees on the Zoom, take a close look and do a little reading Saturday if you don't have anything else going on, because this is really, this is fundamental, like to understand how surrogates are related to diseases and interventions and outcomes and how we can be misled is really, really crucial. And the billion with a B, the $1 billion question here is, do we have reason to believe that amyloid is a valid surrogate? And there are some issues. Now, the issues with amyloid are complicated, okay? So there are some reasons that prior candidates have failed. Biogen will tell you that. We studied people too late. We studied people who we didn't know had elevated amyloid. We didn't have a drug that lowered amyloid levels enough. Group level correlations appear to be more persuasive. I'll come to that in a minute for the biostatisticians in the group. And other products in development, per the FDA and their reviews, other products may provide evidence, an early suggestion of correlation. So maybe amyloid is a good surrogate. And yet you have this systematic review published in the British Medical Journal. I believe it's BMJ Open, or maybe it's BMC. In any case, you can find the article. But the point is, there's a published article. I think I'll let you read the conclusion for yourself, but you don't have to have a PhD in epidemiology from Hopkins to read this and wonder whether amyloid really is a valid surrogate or not. This is so important. And I just want to share with you one table that totally confuses me. And, so how do we know if something is a good surrogate? Well, it would be good to see analyses of correlation that are convincing. Mediator analyses are important, right? And in this case, actually, the FDA's biostatistical review suggests that the sponsor's mediator analysis was not statistically significant and was nominally greater for low dose than high dose product, even though the low dose product didn't work in either trial. And then meta-analyses, right? So these are at least 3 methods that we use epidemiologically, biostatistically, in order to understand the validity of a surrogate. So correlations, mediator analyses, and meta-analyses. So which of these is the right one that we should be looking at? Because if you look at the last column, the correlations range from 0.99, which must taste pretty good to Biogen, to negative 0.04, which would raise a little concern, right? So I guess one point that I want to make, and I will talk about the road looking forward, but this is just not to have some sort of closure or consensus or greater analysis that pulls this together and really puts a ribbon on it is just striking to me. And we're in a setting where the stakes are so high. And just to be clear, these are all derived from the FDA reviews. Some are from the Office of Clinical Pharmacology, and some are from the Office of Biostatistics. But I'm just sort of highlighting this as one example of many, many, many within the reviews where there's marked discordance between different parties within the FDA. And I think at the advisory committee meeting, I said it's like the sound and the picture are out of sync on an old TV or something, but it's just hard for me as a sort of, educated layperson to kind of figure out and make sense of. All right. I know I've been going pretty fast, but it's because I want to leave time for engagement with all of you. So I'm gonna continue sort of at a fairly decent clip. So let me just come back and say that, you know, for those of you reassured by the fact that you've read that Biogen's going to do a quote unquote confirmatory trial by 2030, that's a long time to wait. If you're a patient or family member with Alzheimer's disease, or an epidemiologist or regulatory science expert who's concerned about what's happened. And lest you be misled to think that a confirmatory trial puts things to bed, think again. So in this analysis of 93 confirmatory trials, note that only note that in only slightly more than half was there a confirmed benefit. And in many of those cases, the confirmed benefit was not actually a clinical outcome. It was a surrogate outcome, either the same one that had been studied pre-approval or a different surrogate outcome. So the label, I mean, there's a whole nother sort of morass if you're looking for, inexplicable decisions by parties involved, where the label initially didn't restrict to mild disease, yet that's the population in which the product was studied. There was no restriction to people with demonstrated elevated levels of beta amyloid, and yet half of people with mild cognitive impairment, 50%, may not have elevated amyloid levels, and as many as a quarter to a third of people with Alzheimer's disease don't actually have elevated amyloid levels. That is, they may have, vascular dementia or frontal lobe, frontotemporal dementia or something else, but Lewy body dementia, but not actually Alzheimer's dementia. So I want to just highlight, these questions and just give you a minute to look at these. And these are the types of questions that I'd be interested in your thoughts in, that we can talk about together. Some of them we've already spoken about, like what to do about such a small magnitude of effect. It's unclear where the responder analysis is, by the way. Where's the analysis that shows the actual number of people meeting a certain threshold of response? What about equity? In a setting where the drug may cost thousands, tens of thousands of dollars a year, and where out-of-pocket costs may be as much as 20% copay, there may be up to a 20% copay in some settings. I mean, you better believe that this ain't going to be similarly accessible. What about all the other things that are required to get the drug? MRIs, a PET scan to demonstrate elevated amyloid, access to a center that will infuse it, transportation, and so on and so forth. So I think that there are lots and lots of concerns here. What are clinicians and patients going to do? Like, how are they navigating these waters? Some of you may have other questions, and I'd welcome you to put them in the chat. We'll get to them shortly. Genetic counseling is a whole nother can of worms. So it turns out that the adverse effects of ARIA, the brain swelling and microhemorrhage, are significantly higher if you're ApoE4 positive. So ApoE4 is a genetic risk factor for Alzheimer's disease. So suddenly you have the prospect that clinicians are going to be thrust into doing genetic counseling. I mean, if you come in and you say, my sister has bad Alzheimer's, well, it can't be too bad, or I wouldn't offer her the treatment. I'm speaking as a generic what I would do, not Dr. Alexander. But the point is, if you came in and said and told a prescriber that your sister had Alzheimer's, part of the counseling about and would otherwise qualify for the treatment, wouldn't you need to know if the difference in the likelihood of this adverse event is 25% versus 40%? Those numbers are approximations, so you would have to check the actuals, but I don't think that they're that far off. I think that the risk of ARIA is, maybe up to a third of high dose respondents, but it increases by at least a third, I believe, if you're ApoE4 positive. Most people with ARIA are asymptomatic, by the way, but maybe 10% have symptoms. The symptoms are actually symptoms highly overlapping with progressive Alzheimer's disease: falls, confusion, memory impairment, fatigue. So and then 1 to 2% have severe symptoms. It may require hospitalization, intensive care unit admission, and the like. So lots and lots of questions here. Interestingly, in this analysis of who would satisfy or qualify for the treatment, this analysis that was recently published suggested that more than 90% of Medicare beneficiaries with Alzheimer's disease and more than 85% of those with mild cognitive impairment may not qualify for the treatment, which may be one of many factors that helps to explain why very, very few people are actually receiving this currently. This is not, this is also a really important and I think well argued piece, which essentially argues that new interventions don't just have sort of technical dimensions, they have economic and moral dimensions. And I think that, if you start thinking about a family that's struggling with this common and often devastating disease, I think it will give you pause to consider the varied ways that this treatment may or may not actually affect their lives. Everything from the potential for all of the testing, the MRIs, the PET scans, the transportation, the risks of adverse events, the potential costs of the treatment, the genetic counseling and all of that sort of being layered in, to say nothing about the opportunity costs. There are millions of patients and caregivers that would benefit from tailored education and training programs that have already been demonstrated to have benefits for this disease, arguably in many settings much more convincing than Aducanumab. So I'm talking about memory cafes and collaborative care models and REACH programs, which are resources for enhancing Alzheimer's caregiver health programs, and they're not getting these. So what will be the implication of this on access to or use of those other treatments, to say nothing for how this may affect drug development and the like. Maybe the FDA credibility is also taking a hit. So these are unpublished data that we have where we did a national poll, a national household survey or national poll, and look at the change in beliefs about sort of confidence in the FDA's in the FDA's sort of mission focus and ability to serve the interests of the American public before and after consideration of the case of Aducanumab. As with any study, the study is just one picture of the elephant, if you will, and has to be taken into context in terms of how the questions were posed and the like. Nevertheless, I think that there's a real question about how this has impacted the confidence of the public in the FDA. And I should say, I'm surprised it took me so long to get to this point in the talk, but I should say it's a privilege to work for the FDA. I've been fortunate to engage with the FDA in many, many contexts over 20 or more years. And it's a remarkable institution. And there's a reason that the FDA is widely regarded and well regarded and looked up to around the world for the quality of the science and the quality of the regulation that they do day in and day out. So I just want to be clear, what makes this case so striking is not that this is business as usual, which the FDA has said, but which couldn't be further from the truth. It's that this stands in such stark contrast to business as usual for the FDA. And it's just puzzling how this could have occurred in the context of the care and caution and prudent regulation that the FDA exercises on a daily basis. All right, so again, for Saturday reading, for those of you interested, this was a piece that we published that discussed the views of the advisory committee after the fact. So I will leave it to you to go read that in your infinite spare time. I've already told you that there's been enormous pushback. We have 4 different investigations: House, Senate, FDA, and Office of Inspector General. The VA won't cover it. Harvard Pilgrim says it's experimental and investigational. Cleveland Clinic and Mount Sinai ain't offering it. And so this leads to the first live question poll. And I want to play, while Taylor is posting the poll, which you can release now, Taylor, I want to say there is a counterpoint, which is, consider the magnitude of unmet need. Consider the patients and their caregivers who have been waiting for 2 decades for new treatments. Consider that that the argument that it's not just up to the FDA to decide that that patients should have access to a treatment, that patients and caregivers can decide on their own or with using supports as they wish. Consider the ways this may put wind in the sails for the venture capital markets and drug development. Clearly, I gave my push and my counterpoint a little late, but we'll give you another minute or two. There's 78 folks, so it would be great to know that at least 60 are in front of their computers now. We're up to 46, 47. A few people are coming back from their multitasking and working on those manuscripts. 53, 55, 56. Just need 1 or 2 more and we'll move on to the next poll. And just so I tee it up, perfect. So you can see these results, or if not, whoops, let's end. Taylor, we can share these results. I'm not sure that everybody can see them. So it's so about 50 out of 60 felt that this was a question about formularies. I'm sorry, Hopkins formularies rather than FDA approval, but about 50 out of 60 felt that Hopkins should not offer this product. About 10 or 12 out of 60 felt that Hopkins should offer this product. So we have one more that we will release now, which is putting on your clinician hat. I know many of you are not clinicians, but imagine that you were a clinician. Or what would you think that your doctor should do? So let me read this. Take a minute to think about it. So imagine Aducanumab is on formulary and someone who would have qualified for the trial asks you whether it would be suitable for them. So you're a nurse or a doctor or a pharmacist. Well, let's say you're a prescriber. So you have prescribing authority. And someone asks you whether they qualify for it. And they say, and it's on formulary, so you can access it, and they ask you whether it would be suitable for them. So would you discuss the risks and benefits and let them make the call? Would you decline to offer it because of uncertainty about its risks and benefits and say, I just don't think I can offer it because we just don't know enough? Would you decline to offer it and refer them elsewhere? Say, I know Hopkins, it's on Hopkins formulary, but I'm going to refer you to University of Maryland. I just don't, I'm just not comfortable making a decision, but Maryland has a great Alzheimer's center. Would you advise against it yet offer it anyhow? Would you say, I just really don't think it's the best idea, but if you really want it, I'm not gonna prevent you. So against my better judgment or sort of begrudgingly, I offer it. Or would you advise for and offer the product? Would you say, I can understand why you'd want it, and we'll monitor you closely, and I'm happy to offer it for you. I'm happy to sort of make the decision on your behalf. A little bit of a harder question. I know there's 60 of you at least, or 61 that are in front of your computer. So how about the last few of you? And so this is interesting. So I would argue, so we can release the poll. And so essentially most of you say either that you would discuss the risks and benefits and let them make the call, or that you would decline to offer it. And I would argue that this first choice is illusory. And the reason why is because, as a clinician, there really is no fully neutral option. So I can see why that would be an appealing choice. And I don't blame you for choosing it, for the 36 of you that did. But there's really not a neutral option when you're in a room with a patient. There's no real full sitting on the fence. I mean, if nothing else, your affect is going to reveal your beliefs about it, and so your body language, just everything. So so this is a really interesting one. All right, so let me wrap up. I actually understand that Hopkins, I'm told, has declined to cover this on formulary. I don't know that that's true, and I haven't seen any fanfare or public announcement about it. So some of you may know more than me, and if you do, please feel free to add it to the chat. But my understanding, what I heard secondhand, was that Hopkins had decided not to add it to formulary. All right, so there is more coming, I promise you. So Lilly has a product under the same pathway, accelerated approval, Alzheimer's disease, predicated on reducing amyloid. So this is not a play that has ended. And here are just 3 areas where I think we can learn from this and improve our system going forward. So I want to wrap up, so I'm not going to spend a lot of time on this, but there have been some very thoughtful suggestions, for example, regarding how to improve the integrity of accelerated approval, such as by only granting accelerated approval after a protocol has been written and a confirmatory trial has begun enrollment, which would incent manufacturers to really get those confirmatory trials done. Another suggestion is that accelerated approval should automatically terminate if the confirmatory trial is not complete within 4 years. So this was a suggestion Zeke Emanuel recently made, and I think it's an interesting one. That's the median, that's longer than the median length of time for confirmatory trials examining cancer treatments. And that too would provide additional incentive to get these trials done and get them done well. Another would be to modify pricing. This is beyond the purview of the Food and Drug Administration, but why should we be paying the same amount for a drug that doesn't have confirmed clinical benefit as a drug that does, holding all else equal? So that would be an opportunity for example, CMS to with probably some statutory change in legislation to pay less for products that haven't been demonstrated to be safe and effective with respect to clinical outcomes. All right, so I'll let you take a brief look at concluding thoughts. And, you know, I think the most important one I'll highlight is that this case offers a glimpse into the field of drug development and regulation, highlights the value of people like you and me, epidemiologists, biostatisticians, and trialists to the scientific and regulatory community. So very much looking forward to your thoughts and reactions. And Taylor, I thank you for your assistance, and like to thank Janet and others that were involved in welcoming me, and look forward to engaging in the next 15 minutes. So I took 5 back, but I still am giving you 15, and you know, in the weeks and months to come. So thank you.
Thanks, Caleb. I think we can start with the questions in the chat. Let's scroll up here for a bit. So the first few were regarding the trials that you covered. Is the placebo getting any worse progression of the disease that we saw?
Yeah, well, so the placebo, so I don't know that we fully understand why there was this worsening of placebo over time. And so I don't know the answer to that, Janet. I think it's a great question. I guess I feel like I'm only educated enough about these matters to understand that this was something that was raised and flagged by the FDA biostatistical reviewers and raised a lot of concern. And just to be very clear, the biostatistical team at the FDA did not support approval, and in fact has very, very substantive criticism against approving the product based on the evidence that was brought to bear. So, but it's a great question. If I can, Sharisa, I'm sorry, go ahead, Doug, and then I know Larry, I see Larry's hand raised. That will give me a chance to read through some of these also that are in the chat, but go ahead, Doug.
So Caleb, as regards your question about an accelerating disease course, I think there are several examples of that when you get to late stage disease. And, you know, I'm not sure I'm enough of a neurologist to know the answers here, but it seems to me amyloid may be a relatively late marker. The failure of the amyloid therapies consistently suggest that we indeed are intervening too late, and that amyloid may not even be a very good target. It may merely be a marker for disease, as you know. But accelerating disease course over time, we see this. We see this in a variety of diseases. And so I am not at all surprised that the placebo group got a lot worse in the second year than it did in the first. And to expect linearity, I think, is to be naive.
Yeah, I guess I thought, and I may be mistaken here, Doug, but I thought that this was looking at the sorry, let me just share the screen again. So I think that this is looking at the two different groups of people, but it's looking at them both at the same time point. In other words, it's looking, if you look at the x-axis here, it's looking at both groups at 80 weeks. It's simply looking at people that were enrolled early and followed for 80 weeks versus enrolled later and followed for 80 weeks. So I don't think that, I may have misconstrued what this represents, but I don't think this can be explained away by the fact that the disease gets worse over time. Larry, go ahead.
Yeah, Caleb, great talk. 18 months for a clinical outcome, and it seems to me awfully brief. And I know I think you made a comment as though, well, if we waited another 6 months, and I think that was more, dealing with sort of, additional peaks. But it seems to me that perhaps because of the patent, cycle, that 18 months might be selected, but quite frankly, 3 years or longer, or something that. But I can just tell you, I chair a DSMB for a trial of cognitive decline in which it's a nutritional intervention, and they go at least 3 years. So I look at it and I go, yeah, maybe the effect size was small because, they pre-specified 18 months and really should have gone 3 years or more for a study of cognitive decline.
Yeah, that's an interesting comment, Larry, and I don't disagree. Would you argue that any future trials for Alzheimer's in this type of setting for drug development should go longer? And why do you think the FDA settled for 18 months, or why do you think the FDA and the sponsor settled for that, if based on your insight?
Yeah, I can't, but I can also say that maybe this follows up on what Doug said, that we know that this is a very progressive and slow, and maybe at the end it escalates, faster. But I've been, making the case that I'm not sure we'll ever have trials that are sufficiently long given the time course. And also because we need to start earlier in life, when people. So I've actually been a pretty big skeptic of trials of drugs for cognitive decline, and certainly ones that are so short as to have a primary outcome at 18 months.
Yeah, thank you for that. One, I'm looking at some of the other comments, and Sean, I'll speak to yours, Sean Diffenderfer, about the variability within clinical, can I speak to the amyloid deposition variability within clinical trial participants contributing to differences in outcomes between trials. And there was a lot of effort after the fact to understand how particular subpopulations of patients may explain the differences in the outcomes among the trials. And then, of course, as I showed with the correlation analyses, whether or not you look at sort of individual level correlations or group level correlations and sort of the other assumptions of those analyses, you reach markedly different conclusions about whether or not there is even a relationship between amyloid lowering on the one hand and cognitive improvement on the other. So what I can say, Sean, is that there were no post hoc analyses that are convincing that we understand the basis for the difference between the two trials. In other words, there were no characteristics of individuals that appeared to benefit versus those that don't. that appeared to predict the likelihood of response, nor were there any discernible baseline differences between the trials. Maybe this is another way of saying the same thing, but it was not clear that there were baseline differences of the trials that could explain why one reached such different conclusions between these two.
So, Caleb, can I throw out a straw man here?
Yeah.
I would argue, or at least would try to argue, were I a debater, that the reason is that aducanumab is ineffective, that the very minimal differences you see, although consistent across subparts of the scale, are probably collinear, and that if they do a third trial, they'll find it's ineffective when you see it only in one subgroup and not replicated in the other. Consider that as a possibility. We've seen that multiple times in drug development, where it works in one subgroup and they go and repeat a study use in that subgroup and it doesn't work, and so forth. So just at least one potential interpretation is that you have no evidence that this drug works other than a minimal statistical difference.
I thank you. Mariana, so Kyle has a question about pricing. So the drug is priced at, I think, $54,000 or $56,000 is the list price. ICER, the Institute for Clinical and Economic Review, has a value-based price of, I think, $3,000 to $8,000. And so what do we know about the decision about how to price this drug? I think there's lots of opportunity for good work. What you have is a world of difference between what Biogen has decided, how they've decided to price it, and what we think, and what a very good nonprofit institution believes is a value-based price. So, more will be revealed. I don't have any, I think this is one window through which to view current debates about drug pricing and ask how our system got so dysfunctional. But Mariana, if you have anything else you want to add, please feel free.
Thank you for answering my question. My thought was, is the price reflecting the uncertainty around this clinical effectiveness and safety, et cetera, or is it the price more reflecting a groundbreaking, the aspirational side of the company to be a groundbreaking new therapy for Alzheimer's? And from what you describe, it kind of seems like the second case. And then the question would be, why didn't they shoot for an even higher price as the trend that we have been seeing with other drugs?
That's a fascinating question, and of course, while we don't have the counterfactual, it's a great question. Whether should we be thankful for 54,000 or 56 because it's only 54 or 56,000, but if the drug really worked, then ... I mean, if we had a lot of confidence about it, so I don't know. We'll have to see. Karen, you have a question, Karen Robinson, about who needs to change the FDA process and who and how can these changes be made. And, I think there is a lot of scrutiny now on the accelerated pathway above and beyond aducanumab. The OIG report or decision to investigate was interesting because it's sort of using aducanumab as one case study by which to review the accelerated pathway. And, the FDA has lots of regulatory flexibility that doesn't require change in statute. And then there are things that might require change in statute. I'm of course not an FDA lawyer, although I like to hang out with them. But I think the sorts of suggested changes that have been made at the FDA are, in addition to doing a really thorough postmortem and figuring out how the heck the train got so far off the tracks, and is this an episode, is this an instance in the worst case of regulatory capture, where the FDA failed to, keep essentially the FDA forgot that they were the umpire, right? They weren't the Astros or the Braves, they were the umpire and they forgot that. But other suggestions regarding the use of endpoints. I mean, that the FDA should only be using surrogates that have much more evidence than amyloid does and better explaining the rationale. There needs to be greater transparency regarding recruitment and sort of some of the regulatory processes and trial conduct for trials that are as consequential as these. Gary, looks like you may get the last query and then I the last comment. So Gary Rosner, please.
So thank you for that. So I have a question about the acceptance of this as a surrogate endpoint, because I think you presented information or data from other studies that showed that treatments that were not effective clinically had a strong effect on the surrogate, which violates one of the criteria that we statisticians would use for declaring something a surrogate. So I don't understand how it got that far. Do you have any added insight that you could share?
Yeah, it's a great question, and I don't have much. I mean, I will say that it is striking for as consequential, again, for as consequential a decision as this is, it's remarkable to me that the FDA wasn't able to speak with one voice and put in one place the totality of evidence that they're relying on to use amyloid as a surrogate. And I think the reason is partly because the decision was only made at the 11th hour to rescue this product from the jaws of defeat. In other words, it only became clear 97% of the way through the drug's development that it wasn't going to be able to get over the standard approval hurdle. And so what you have is a lot of disparate information and conflicting information from different FDA parties that's been placed in the public domain, but there's been no cogent single analysis that I can point you to that's really ties this together and puts a ribbon on it. You have lots of conflicting information about mediator analyses, about correlations, and so on. So thanks for the question. And I think we're out of time, but I again want to thank everybody for the opportunity to share a little bit about this continuing tale and for the privilege to work in such a wonderful environment and to work with all of you as colleagues and trainees. So thanks again.
Thanks, Caleb. This was a very insightful talk. Appreciate it.